Thank you for having me present some of my data and findings on. So my name is Dr. Victor Dieriks, and I'm a senior research fellow at the University of Auckland in New Zealand. And I want to talk to you a bit about my research and my findings. So my main focus has been Parkinson's disease for the last 8 years, but over the last couple of years, I've been looking more in detail into multiple system atrophy. So you'll see a mixture of Parkinson disease data and multiple system atrophy data. So to start off with the obvious, Parkinson's disease is a progressive neurodegenerative disorder of the central nervous system affecting the dopaminergic cells in the substantia nigra. A very difficult definition. Basically, it says it's an ongoing disease that affects the brains, and specifically, it kills off a specific neuronal population in the brainstem of the brain. And when those die off, we see the typical motor symptoms. What's very characteristic in Parkinson's disease is the presence of these Lewy bodies. They sometimes look like a donut shape or round. These are Lewy bodies or Lewy neurites. These bodies and neurites consist primarily of alpha-synuclein, which is a vital protein in Parkinson's disease and also multiple system atrophy. So Parkinson's disease was first described in 1817, and unfortunately, since then, not much has happened to cure or treat Parkinson's disease, especially not targeting the underlying symptomatology. So when we look at multiple system atrophy, the definition is quite similar in that it's a rare condition of the nervous system that causes gradual damage to the nerve cells of the brain. It also results in motor symptoms, neurogenital dysfunction, and cardiovascular dysfunction. Typically is that we see Lewy bodies and neurites containing alpha-synuclein in neurons, but very characteristic in oligodendrocytes. And oligodendrocytes are probably one of the key players in multiple system atrophy, which is something we don't see in Parkinson's disease. So this graph, it's a bit confusing and it's supposed to be this way. So pathologically, Parkinson's disease and multiple system atrophy are grouped together with dementia with Lewy body disease as synucleinopathies. So they all have different causes, but they're all characterized by the abnormal accumulation of alpha-synuclein aggregates. So the problem is that all of these sub-diseases have overlapping symptoms. And each patient within these sub-diseases reacts differently. They have different symptomatology, and the symptomatology overlaps between these sub-diseases. So it's really hard to distinguish between the diseases. And that is something that is really common with multiple system atrophy. At the early stages, it's most frequently confused with Parkinson's disease and vice versa. On top of that, you don't just have alpha-synuclein protein aggregates that form in a brain. Sometimes we have brains with multiple types of protein aggregates. So a study done on Alzheimer's disease found that in 38% of the cases, they had 3 different protein aggregates. And 12% of the cases, they had 4 different types of protein aggregates. So you can imagine that each of these protein aggregates influences the brain, but if you have multiple ones, they each individually influence the brain differently. So it's a very complex disease, which makes Parkinson's disease and multiple system atrophy really complex to study. Ending us with this after 200 years of research, but still there's quite a bit of stuff that's happening, and we found a lot of information. So we know that Parkinson's disease is caused by a complex interaction of genetics and environment. Exposure to metals, pesticides, bacteria, and viral infections are known to be linked to Parkinson's disease. And in case you're wondering, infection with COVID can also cause the sudden onset of Parkinson's disease symptoms. In 2020, about 5 different case studies appeared. So within 2 to 5 weeks, after infection with COVID, these people started showing Parkinson's disease symptoms after treatment with the Parkinson's disease medication, L-DOPA. Their symptoms subsided. The issue will be what will happen over the long term, and that's something that we don't know yet. So up to now, we've been looking at Parkinson's disease as the same disorder. Multiple system atrophy was kind of like linked within that. But it's actually a combination of a range of sub-diseases that have these overlapping symptoms. There is one thing in common, and that is in all these sub-diseases, we see the presence of protein aggregates. So the accumulation of alpha-synuclein, which has its normal function, aggregates into, over different stages, into these large protein aggregates. And the formation of these protein aggregates is toxic to the cells. As the disease progresses, these aggregates can move to neighboring cells where the process is repeated. Over time, more aggregates are formed until the cells can no longer cope with it, and it leads to cell death. So when we look at Parkinson's disease at the different stages, we see that the alpha-synuclein aggregates, the alpha-synuclein pathology, is found within certain areas at the start. Most commonly, it's found within the olfactory bulb, which is responsible for the sense of smell, and in the brainstem. So as the disease progresses, we see that it migrates to neighboring cells, and towards the end of the disease, the entire brain is affected. For multiple system atrophy, the olfactory bulb is not as much affected. And unlike Parkinson's disease, MSA is considered more to be a sporadic disease, so not that much linked to genetics. That is not to say that genetics do not play a role. There have been certain genetic variants of the COQ2 genes that have been shown to play a role in Asian populations, but then this did not show any differences in Caucasian populations. So I've been talking about this accumulation of damage here. Initially, cells can cope really well with these formation of aggregates. They can even break them down, and they can clear them. But at certain points, the damage will outweigh the repair mechanism, and aging and cellular stress accumulates on top of this. So in a healthy person, we see that over the course of their lifetime, that brain damage gradually increases. And aging is known to be the biggest contributor for Parkinson's disease and multiple system atrophy. But in this person, it never reaches the critical threshold for damage to occur. So when we add genetic risk factors to it, we see that this person reaches this critical threshold and symptoms occur in this patient. The same thing can happen when after exposure to various forms of environmental factors. They don't all have to occur. It can just be one, or it can be a combination. And again, this person starts showing Parkinson's symptoms at a certain age. When we combine both genetic and environmental factors, these people can have Parkinson's disease at a much earlier age. And to give you an example of how bad this can be, in New Zealand, we work with a patient who was diagnosed at the age of 18 with Parkinson's disease. This person started showing symptoms from the age of 12. It took 6 years to discover that he had Parkinson's disease. But then again, this is just to say that the age of onset can vary quite dramatically between these patients. And it depends on each individual combination of risk factors. That being said, we can influence the environmental factors, and we can actually delay the onset of Parkinson's disease to a later point in time. By drinking coffee is one well-known effect. So coffee is known to delay the onset of Parkinson's disease, but healthy lifestyle and exercise play a really important role. And I can't emphasize exercise enough. So exercise does not only delay the onset of Parkinson's disease and multiple system atrophy, even at the point of diagnosis, it will delay the decline of the onset. of the symptomatology. So if we exercise regularly, we will see a slower decline of um, so what is my aim? So we have this occurrence of this protein aggregation uh, in Parkinson disease and multiple system atrophy, two protein aggregates, which causes damage to the cells. My aim is actually to break down these protein aggregates and to bring them back to the functional protein, either by cleaving and removing these protein aggregates or by untangling them and returning it back to their normal function. By doing so, we're reducing the total amount of toxic aggregates within the cell, and we're reducing the amount that can spread to neighboring cells, thereby delaying the spread to neighboring regions and delaying the onset of the more severe symptoms. My research is not aimed at curing Parkinson's disease or multiple system atrophy. The disease is just too complicated. My aim is to delay the disease by a year, 5 years, 10 years. And if we combine it with other treatments, we can even delay it by 20 years. And I like to compare this to HIV research. In the 80s, HIV was considered a deadly disease. But now, with the combination of various treatments of antiviral drugs, people can live a healthy life of 40, 50 years. So the same thing I want to do with Parkinson's disease research and multiple system atrophy research. So we recently discovered that not all aggregates are created equally. So depending on the original insult, the cellular environment of the various cell types can change differently. So we can change the acidity of the cells, the salt concentrations, and these conditions lead to unique 3D conformations of alpha-synuclein aggregates that we call strains. Once a strain is formed, the structure does not change and it functions as a template so that subsequent aggregates will have the same structure. To make this analogy a little bit easier, compare strains to different types of pasta. Pasta always contains the same ingredients, but depending on how you make it, you either have spaghetti, linguine, macaroni. So even though the um, the ingredients are the same, the outside looks quite differently, and how it tastes and how it interacts with the cells will be quite differently. So here I have two different strains: the fibrils, which it resembles spaghetti, round and long, and ribbons, which more resembles a linguine, which is flat with a small twist. So when we inject fibrils into rodents, we see a typical Parkinson's disease phenotype with these Lewy bodies in neurons. When we inject ribbons into rodents, we see a mixture of Parkinson's disease and multiple system atrophy phenotype, with the typical feature of oligodendrocytes containing these protein aggregates. So we identified in the lab which cell types contain alpha-synuclein aggregates. And then we painstakingly looked at each individual cell in a particular area of the olfactory bulb, which is responsible for your sense of smell, to see which cells were affected and how many cells were affected. So we have different cell types within the brain. Everybody knows neurons because they transmit all the signals. Oligodendrocytes basically insulate these neurons and help facilitate more efficient signal transfer. Oligodendrocytes are star-like cells, which are involved in information processing and glial scar formation. Microglia are the cells that are involved in the resident immune cells of the brain, so very important in protecting us against infections. Parasites are a bit of the underdog in these cells. They regulate blood-brain barrier function, but are also important in inflammation and are lost in Parkinson's disease as well. So this graph took about 2 years to make, with 4 people working on it quite intensely. And it shows the amount of cells that contain alpha-synuclein aggregates. And here we see that microglia, parasites, and astrocytes all contain alpha-synuclein aggregates. Oligodendrocytes didn't contain any, which was not unlogical because these were Parkinson's disease olfactory bulbs. And of course contain alpha-synuclein as well. So this shows that it's not just neurons that are important in alpha-synuclein. Non-neuronal cells such as these microglia, parasites, and astrocytes are important as well. So next we look at how can alpha-synuclein spread. So there's different mechanisms that cells can use to spread alpha-synuclein. The one thing that I looked at is tunneling nanotubes. Tunneling nanotubes are small connections that are made between cells and that allow exchange of materials between cells. So here you have two cells, they contact each other, and as they move away from each other, some parts of the cell are still connected. And these small connections allow for exchange of proteins like alpha-synuclein, but also viruses and other materials to exchange between the cells. It's not an efficient way of transport, but as we all know, Parkinson's disease and multiple system atrophy can take years to decades to eventuate. So even a slow process might be important in spreading alpha-synuclein. So to give you an example, here is a video of two parasites with the formation of tunneling nanotubes, and you'll see the transfer of material from one cell to the other. So just to show how these tunneling nanotubes are able to transfer material between one cell to the other. So one of the unique things that we can do in the lab is we can grow neurons and other cell types from brain biopsies. So in the lab, we took this biopsy, we took out the cells and we put them in this petri dish. So this contains neurons, astrocytes, microglia, oligodendrocytes, and parasites. What I did here is I added fluorescently tagged alpha-synuclein. And you'll see that some of these cells become really red. And these are the cells that are taking up the majority of the alpha-synuclein aggregates. And in this case, these are all microglia. So as I mentioned before, microglia are the immune cells of the brain, and they take up foreign material. And in this case, it's the alpha-synuclein aggregates that they take up in mass. But they don't do a 100% efficient job. So you see here that this is a neuron in red here, and there's one little spot, in this case green, which is alpha-synuclein, which has been taken up by this neuron. So even though the microglia, which is one that's just here, has a lot of alpha-synuclein taken up, some of it does go to the other cells. There's a lot more within this parasite. You can see quite a bit more alpha-synuclein aggregates, and of course, most of it is in the microglia. But this is to show that not just microglia are important, it's to show that parasites also play an important role. So we know from previous research that astrocytes and microglia are involved in a combined attempt to clear as much excess alpha-synuclein aggregates from the cells and from the brain. And they do that in an attempt to prevent Parkinson and MSA pathology to spread to neighboring cells. So why am I using parasites? Well, parasites also contain these alpha-synuclein aggregates, as I've shown you. They're lost in Parkinson's disease. They're involved in spreading of alpha-synuclein, but unlike microglia and astrocytes, we are able to grow parasites from dead brains. So in Auckland, when a brain comes into the brain bank, we're able to take small parts of the brain and culture the cells in the lab. So as such, we have the Hugh Green Biobank at the university, and that contains over 270 different parasite lines from different human brains. So we have Parkinson's disease, Alzheimer's disease, and the beautiful thing about this is we can grow these cells, we can freeze them down, store them, and when we need them, we can do experiments on them. Parasites characteristic specific to the brain that it originated from. which is not something we can't do with astrocytes and microglia. So what have I been doing? So I've taken parasites. So here's an example of some parasites, put them in a petri dish, add different types of alpha-synuclein strains to them for 4 hours, and then look at various time points afterwards and see what happens. I use 5 different strains: fibrons, ribbons, which is associated with MSA, and fibril 65, 91, and 110 that I've been supplied to with my collaborator, Dr. Ronald Melky from France. So here's an example of pure parasite culture, just parasites that take up alpha-synuclein aggregates and are able to accumulate them in certain cells. So you see that some of these cells do not contain any alpha-synuclein. They're managed to clear it, whereas other cells do seem to have a lot more of it. So they take it up really efficiently, but can they also break it down? And that's something that we've been able to show with Western blots. So in Western blots, we can separate proteins based on their size. So compare a Western blot technique to walking through a forest. So the proteins walk through a forest. At the start, the forest is quite sparse, so it's easy to walk through. As you walk through the forest, comes more and more bush that goes in there. So small proteins can still walk through it quite efficiently, whereas big proteins are delayed a bit more. So we see a separation. And that is something we've done here. So I've added alpha-synuclein aggregates to the parasites, and we only see this band, this size of the alpha-synuclein. After 4 hours, we see the original size and some smaller fragments. 24 hours later, we see that the parasites have broken down the alpha-synuclein, and we only see the smaller fragments here. When we do the same thing for ribbons, we see the original bands. 4 hours later, after adding it to parasites, we see smaller bands. And again, after 24 hours, we only see the smaller bands. Other strains show a different pattern, but I won't go into detail here. It's just for this, it's important to see that parasites can break down this alpha-synuclein. So they break down alpha-synuclein. We see it in the brain that parasites contain alpha-synuclein. So what does it do with these cells? So for this, we did RNA sequencing of 6 different parasite lines from different cases, and we treated them with the different strains. So you might be asking, what is RNA sequencing? So imagine that all the genetic information that is within your body is printed in books, and these books are stored in a library. These books contain the recipes to make everything that is needed in your body. Alpha-synuclein, the proteins we need to make muscles, antibodies, the color in your eyes, everything is stored within these recipes. Now imagine that you walk into this library and you see books in the shelves, and you see books that are open on the tables. Well, these open books are the ones that contain the recipes that are needed at that particular time. So with RNA-seq, we walk into the library and we basically take pictures of these open books. So we know at a particular time which recipes are being used. And we do this by comparing cells that are treated with these strains, and we compare them to cells that are not treated. And we see the differences in genes that are used versus the ones that are not used. And this allows us to identify out of thousands and thousands of genes, which ones are being used and which ones are not being used. And we see for the case of fibrils, about 170 of these genes are differently changed when we add alpha-synuclein versus if we don't add the alpha-synuclein fibrils. So we've done this for all 5 of the strains. And what is quite unique is when we overlap these different changes of these 5 different strains, we see that none of the genes occurs for all 5 strains, which means that even though is always at the core of it, it's the shape that makes a big difference in how cells interact with these strains. So it's really important to look at these individual strains because these strains make a big impact of how the cells react. So subsequent treatments should take into account the strain type. So what have I been doing? So I've been using these parasites, as I mentioned, done RNA sequencing, we get these different changes. But what's really important is that the next step is that we actually validate the expression of these genes. So with RNA sequencing, it's only a snapshot in time, but we want to make sure that at a protein level, we also see these changes. So we have tested over 100 antibodies, parasites treated with and without these strains. And we've also looked at tissue, brain tissue from Parkinson's disease, multiple system atrophy, and compare them to healthy controls. It's really important to do both of these because in culture, we only study an aspect of the disease, in this case, parasites. Whereas in brain tissue, it's much more complex. We have different cell types, have a 3D structure. So it's really important to validate it on both these systems. So of the 100 genes validated thus far, we found 6 that showed significant differences at a protein level, 2 of which are linked to ribbons, which is the MSA specific strain. We're currently looking at finding additional genes that play a role and validating them both in Parkinson's disease and MSA tissue. So there's a lot to look forward to. So once we have this validation sorted, we will use techniques like CRISPR and RNAi. I'm not going to go into detail in this, but the idea will be to target these genes specifically in order to reduce the protein aggregation or untangle them. So depending on the particular function of the protein that we're changing, thereby reducing the alpha-synuclein loads and reducing the spread to neighboring cells. So where lies the difference? I hope I've been able to emphasize the uniqueness of oligodendrocytes that oligodendrocytes play in multiple system atrophy. So as I've shown you in my Parkinson research, oligodendrocytes were not affected, and but they play a key role in MSA research. Secondly, the strains. The different strains play an important role in either developing Parkinson's disease or multiple system atrophy. And I hope I made it clear that depending on the strains, cells react differently, and these strains can cause either Parkinson's disease or multiple system atrophy. But the important thing will be understanding what leads to these MSA specific strains. And that is still a very complex question that we're trying to answer. So in my lab, we propose that MSA may require insult in both of these factors, being strains and being oligodendrocytes. Thereby, and because we need both things to go wrong, it could account for the rarity of the disease. So multiple system atrophy is a much rarer disease than Parkinson's disease, as highlighted by the incidence here. So individual changes to oligodendrocytes might be insufficient to drive the disease because oligodendrocytes need the supply of neuronal alpha-synuclein to form alpha-synuclein aggregates within the oligodendrocytes. If that is not present, then MSA cannot start. So if we only see changes in the neurons, then this will probably result into Parkinson's disease. But if both combine, then we will have MSA that commences, and we have, as a result, a disease that is affecting the brain much more, and we have a much faster progression. So MSA strain formation and pathogenesis is likely to require this multi-insult hit by altering both neuron and oligodendrocyte function. So this is, in a short, some of the research that we're doing and some of the recent findings and pathways that we're investigating. And I will end it here by thanking everybody that's working in my lab, all my collaborators, and not that make my work possible. I thank you for your attention.